My lab really is a plant physiology lab, so I'm really interested in what's happening with the tree health themselves. So we study general forest health. So what I'm going to be talking about is growth declines associated with the white pine needle defoliation. And when I say growth declines in the context of this talk, I'm really talking about wood. There are a lot of other aspects of trees and growth that are just have intrinsic values in our forests, like ecosystem services and hydraulic regulation. But growth is really kind of the economic driver of some of our forest management. So that's what I'm going to focus on today. So just a little bit of background to cover some of the basics here. We know that this is a complex of several fungi that are defoliating the needles. Work that Stephen's done has confirmed that this Leishmania seems to be one of the main drivers. We know that this is rains dispersed and they infect through the needle stomata. So the stomata are just those pores on needles and leaves. That's kind of the interface with the environment. So this is where gas exchange happens. This is where CO2 is coming in, water vapor is going out, and photosynthesis is happening. So you can imagine that a loss of crown volume or foliage is just less potential for photosynthesis, and you're going to have less carbon allocated to the different parts of the tree. And we know that these defoliation induces premature needle drop of just the second and the third year needles, while the current year needles stay intact. A typical healthy pine will hold on to those older needles throughout the entire growing season until they have a normal senescence in October. So just like normal deciduous trees like maples and beeches, they'll drop those older needles in October and then hold on to their healthier ones. And then we know that there was a regional outbreak that kind of happened somewhere around 2009. So that's where I'm going to base a lot of my later analysis on, is the assumption that this outbreak kind of happened around that time period. So I have a few research questions that I'm going to address for this talk using two main methods, really. So the first one is litter fall. And what I mean by litter fall is that's just the dry, dead needles that are coming off the trees. So my questions with this is, what is the timing of the defoliation? So can we pin down exactly when the defoliation is happening? And then how much is coming down during each month? And then can we see a difference between sites? So my sample design, I had a geographic gradient that I wanted my sites to be across. So we have this larger network of survey sites that the Forest Service set up. So I've kind of just picked a subset of these to sample for the 2014 growing season. So you can see we have two in southern New Hampshire. One's the Thompson Farm, which is one of the farms on the UNH campus. And then the Fox State Forest, which is a state-managed, mostly pine hemlock forest in southern New Hampshire. And then going north, we have the Massabesic Experimental Forest. This is where Stephen did his spore trap collection. And then even farther north it is in Bethel, Maine, which is a privately managed pine stand called the Chadbourne Tree Farm. And then to address my second part of my question, I'm looking at tree rings, also known as Dendrochronology, okay? So what I'm interested here is, are these trees sensitive to defoliation? Can we pick it up in the tree rings in the wood? And are there declines associated with the defoliation? So to do the litter fall, basically all this is it's a modified laundry basket that I've made, and I've lined with it a mesh, and I've just kind of tacked down within these plots. So for each of those 4 sites, I established 3 30 by 30 meter fixed area plots. And then within them, I just kind of had like this tessellated sample design and then randomly placed the baskets within each of these plots. The plots themselves I picked out based on the fact that they were pine dominated and that they weren't on the edge of anything. So I didn't want any like sort of edge effects happening with my sampling. And then within each of these plots, I also did stem count. So I wanted to know what the relative density of white pine was in those plots. So I just measured diameter at breast height of every tree within that plot, and then based on the amount of white pine in there, I could just normalize my litter fall data based on the difference in density across plots. So here I'm showing the results of that litter fall sampling. On your bottom axis here, you can see my 4 sites. These are actually organized from the southern part to the northern part of my range, which also incidentally happens to be the relative severity that I observed. And then on the y-axis here is the pine litter scaled up in terms of grams per meter squared. So each of those litter baskets, I just collected the pine litter at the end of each month, dried it, sorted it for the pine needles, weighed it, and then normalized it according to the basal area that I found in those plots. And so you can see here each month is coded a different color up here. And so June I've kind of just highlighted as this orange-red color. So you can see that this big pulse event that we get at each site in June. And the other big pulse we've seen is that October one, right? It's interesting to note that the October one is greater for all these except for this Bethel site, where you see a lot coming down both June and July, and then a relatively lower amount coming down when we would normally expect needle senescence to happen. Looking at the same data, I've just grouped it in a different way. So on the left here, we have those 4 sites in the same order, and I've just grouped it by this black bar. So this is the cumulative summer defoliation coming down. So this is what we know is induced by the fungal defoliation. And then the gray bars are the cumulative natural defoliation, so what I just saw coming down in the September and October. And you can see there's certainly a difference across sites. And then just looking at it as a percent on the right figure here, you see that the relative defoliation this summer is somewhere around 50% for even the lower sites, and then up to 75% of the total needles coming down for that Bethel site occurred during the summer defoliation rather than the normal time we would expect. I'll also note that I had initially designed this sample design as to have the Thompson farm as be my kind of control. We thought this was a healthy site, but sure enough we were seeing defoliation there. Stephen mentioned we've been having a hard time finding totally healthy stands. And so one of the next logical thing to do was to see if we had any sort of correlation with what Stephen was seeing with the spore dispersal. And sure enough, this shouldn't really blow your mind, but there was almost a one-to-one relationship between what we were seeing with litter fall and the spore, the spore trap data. So I mean that makes sense. So when needles are coming down, they're infected, they have spores on them, they're spreading. So this is just for that massive bezic site. So why am I so interested in the timing of this defoliation? Well, one reason is that during the summer, those current year needles are still elongating. So you saw this picture already, these 2 branches collect on the same day, and these needles are still really, really short. So what I'm showing on this left figure here is needle elongation. So it's just the needle growth throughout the growing season. And this is at the Thompson farm site. And you can see during the relative time that the defoliation is happening, those needles are only about 50% elongated. They're at about 30 millimeters here, whereas by about the first week of August, they're kind of flatlining on growth and they're at about 60 millimeters. So that's interesting to note because then you have these two twigs, right, if you kind of use this as your reference. If you think about the amount of photosynthesis that's happening on a tree, that has this amount of foliage versus this, there's going to be a big reduction. And then there's also other things that come into play like nitrogen allocation. But I was really interested in how is this loss of basically photosynthetic potential going to affect the wood growth? And then also where is that carbon going? So trees that are experiencing defoliation like this are likely going to allocate more of that carbon towards next year's needle production than this year's wood growth. So to address this, I used Dendrochronology, or as I said earlier, just basically the science of tree rings. So there's several properties about tree rings that I'm sure we all know about. In temperate regions like this, they put on 1 year of growth every year, or 1 ring per year, I should say. And this is just due to the fact that we have seasons, right? So we have a very defined growing period, and then it stops, and we can see that transition in the wood. And then we know that the widths of these rings are kind of indicators of relative growth. So a larger ring is indicative of a good growth year, whereas if you have a smaller ring, that's a year where growth was relatively less. And there's several factors that influence growth, but some of the main ones are just temperature and precipitation, management practices, of course, and then pests and disease. So I'm definitely going to be addressing this disease issue here. So to do this, we extracted tree cores from a subset of trees at each sample site. So a tree core is actually just like a really nifty way to get in there and extract a small portion of the wood to be able to look at the rings without having to cut the whole tree down, right? So we get this small core, and then you can sand it down, and you can see those rings at a really high resolution under a microscope. So for each of the trees I sampled, I got 2 to 3 cores, and then we measured them to just the fraction of a millimeter. And then we converted those ring widths, so if we think of a ring width in like a centimeter, we just converted that to what I'm going to refer to the rest of the talk as a basal area increment. So that's taking into account the diameter of the tree as well. So as the tree grows, the rings are going to get relatively smaller, but area will reflect the total growth. So what I'm showing here on the left is what we call a tree ring chronology, right? So that's looking at every ring through time, and each ring gets a year. So on the x-axis here, we have time. This one's going back to 1970. Most of these chronologies are older than 1970. I'm just truncating it for clarity. Most trees at this site were actually established around the 1930s. There was a big hurricane in the region in 1938. So this is actually a relatively even age stand. And then on the y-axis are these basal area increments. So years of relatively low growth, these are years where conditions were somewhat poor, and then high growth, like we see in the 80s, were somewhat better growth conditions. But I was really interested in what's happening kind of more on this end of the chronology, right? So you can see that these two mirror each other pretty well, but you'll definitely notice that what we're seeing is with the red chronology. So these are the trees that were tagged as high severity, so seeing a large amount of defoliation. Whereas the blue line is this low severity or relatively healthy trees. And what we see on the later end of the chronology is this big divergence where the chronologies seem to mirror each other pretty well up until about this late 2000s period. And so to really quantify this, I looked at the percent change in basal area increment before and after that defoliation happened in 2009. So I was just looking at the mean of 4 years before it happened, so 2005 to 2008, and then after, which is this 2009 to 2012 version. So like I said a minute ago, you can clearly see that there's already lower growth rates in mean basal area increment in high severity trees. So these trees are probably predisposed to something else, which may have facilitated the infection in the first place. But you see after the period, we actually have a slight increase in the low severity chronology, and then a net decrease of 23.8% in that red or high severity chronology. So I'm just going to show you a couple more figures like this from the other sites. Here's Bethel, Maine. So this is where we saw that big defoliation, right? And I also mentioned that this is a managed site. So you can actually see one of the release events here from the 90s when they did a thinning. So you can see that clearly had a positive effect on growth. But we see kind of the same trend in that basal area analysis where we have a slight increase over the period after the defoliation, and then a net decrease of, again, about 20%. And here's the massive music forest. So this one's interesting. Unlike the other two chronologies, these ones don't seem to be predisposed. This is also another even age stand. Mark mentioned there was that big fire in 47. So that actually virtually wiped out this whole forest. And so all these trees kind of grew back around the same time. And I've just kind of put this arrow in for clarity here for 2009. You can see that this is kind of where those two chronologies start to diverge from each other, and they don't see that one-to-one relationship anymore. And interesting, we don't see a decrease in that high severity chronology. What we end up seeing is actually just this big increase in the low severity trees. So this doesn't necessarily mean that the infection is like facilitating better growth of the low severity trees. What this likely is that this has been a good period of growth the last few years for this site, and the high severity trees just haven't really been able to capitalize on it because of the defoliation, and it's really compromising their health. And in order for them to keep up with needle production, they're not able to put on as much wood as those healthier trees. So just in conclusion, to address some of those research questions I posed earlier, we know that the defoliation occurs in a large pulse in June and continues somewhat through July and August. The defoliations differ in magnitude across sites, ranging from about 50 to 76% of the total annual needle drop. The summer defoliations appear to be causing a wood reduction since 2009. And chronically infected trees show a mean basal area increment decline of about 25 to 30% in stands where it's infected. So with that, I'd just like to thank some of my collaborators, my funding through the NHAES Experimental Station and the U.S. Forest Service, and I welcome any questions. Thank you.
Time for one question.
Yeah.
Is there anything we can do about it?
Yeah, so management is definitely the next step, right? So the first thing we had to do is, should we even care? So this was kind of the first work being done to see if there is even a growth decline. And my future work has proposed to actually do some blocked silvicultural treatments, see if thinnings can actually help mitigate the effect. Yeah.
The question I have is regarding the needle length. You talked about the drop is primarily around the second and third year.
Yep.
And yet you're talking about needle length over the course of a year.
Mm-hmm.
Is that, I'm not sure where those needles are growing from.
Right.
I'm confused by that.
Sure. So a healthy white pine will just hold on to its previous year's needles for the most part. It'll drop the most, a lot of the ones it doesn't need. But then beginning in about May in this part of the country, they'll actually initiate a new year of growth from their terminal buds. And so what I was showing with that one figure with the needle length is I was measuring those current year needles. So the length of the second and third year needles aren't changing at all. I was just interested in what were the length of this year's needles while the defoliation of the second and third year needles are happening. It's a bit confusing because there's about a 1 year lag that happens in the cycle. So while those second and third year needles are dropping, those current year needles that are still very small are becoming infected. So it's the fungus is coming in through the stomata. They just don't become symptomatic till the following year, and then those are the ones that are dropped, and the cycle kind of just continues. Yeah.